All right, here's our video, Living Environment Regents, number 10. And we're talking about translation this time. In the last video, we talked about transcription. So let's get a clear page going here. Get our pen ready. We'll start with, I'm going to use white. Use white. Okay, and let me write down, let's write both words. And we're going to talk about RNA polymerase right away after that. So let's go back there. And there's our nitrogenous bases. All right, so actually white would not have worked on a white background, right? So if I write transcription here, so some students ask, well, how can I remember this? Well, with patience, you remember anything, right? There's transcription, and let's put translation on the bottom. So in transcription, so they both have the word trans, and it means in English across, right? In other words, you're going across language, like you want to across language with translation. But if you think about this, they have script. So let's use a different color. This one here, script, means let's read the script first, and then you could translate it, right? Then you can translate it. So you should know what that means. Like if let's say your grandma speaks Italian and she says something, or she writes something, so you read the script first. You're taking that script and then you're translating it to someone else. So that's one way some students have remembered it, or whatever mnemonic device you have used, let me know. All right, so anyway, with transcription, okay, let me draw. Let me get the pen first. We'll stick with green. So this is a very simplified version of the double helix DNA. So in this, we said that there's going to be these RNA polymerase that are going to break apart the bonds, the hydrogen bonds. So let's just say that we had some of these nitrogenous bases, which we talked about in the last video. And let's just say that that was A. Just write anything down. C, C, G, T. And over here, the corresponding A, remember A bonds with T in DNA, C with G, C with G, G with C, and T with A. So, and remember, these hydrogen bonds are broken by the polymerase. And we talked about this in the last video. Okay, and again, I'm doing it very basic here. Change the color. So then we have this RNA polymerase. And we're going to talk about there are three types. We'll talk about that very soon. So it starts at a promoter. The promoter is promoting it, right? In a way you can think about that. The promoter is where it will start, and it will move. It's going to move from the 3' to the 5' like we talked about in the last video. And it's going to read this code, and it's going to create RNA. In the last video, we talked about creating mRNA. So it's going to take this code. It's going to go from T to A, G to C, C to G, C to G, and A is not going to go to T because RNA is going to make uracil with the U. So it's almost an exact copy, except there's no T. It's just U. And like I said in the previous videos, thymine cannot leave the nucleus. If it leaves the nucleus, it will be destroyed by oxygen. And other people have said thymine is in the body, it's very expensive to make. That's another, scientists have also talked about that. So not expensive in the form of money, but expensive for the body. So that's another reason why they said uracil works. So anyway, so here is the mRNA, and it will leave the nucleus, leaves the nucleus, and it finds the ribosomes, and we begin translation. So remember, the first part is transcription. This is transcription. It's reading the code, right? It's gathering the code, the script, and it's going to bring it to the ribosomes so we can translate that. So let's talk about that now. And there are three types of like RNA polymerase: type 1, type 2, type 3 in humans, right? There are others. Plants have a type 4 and a type 5. We're talking actually about humans today. That's us. And of course, we're eukaryotic, and so are plants and other animals. Eukaryotic means it has a nucleus. Let's get a picture of that. So prokaryotic would be like bacteria, it doesn't have a nucleus. Eukaryotic has a nucleus. So a eukaryotic could be one cell or multi-cell. So don't think of eukaryotics are multi-celled creatures. They are, but they can also be single cell. So let's get a picture of that. Right? So prokaryotic, like this bacteria, it doesn't have a nucleus, right? But eukaryotic cells have a nucleus. So that's basically that. And we'll talk about that as we move on to cells in later videos. So there are 3 types of RNA polymerase, and let's break those down. And this is how you can remember them. Let's get our pen going again. Maybe we'll stay with blue. Why not? So if you think about the first part, R, this R is you can think of as type 1, because type 1 polymerase will be for rRNA, which we'll talk about in other videos, ribosomal RNA. Type 2, you can think of this M right here. That'll be type 2. And with the M, you can guess it, it's messenger RNA, mRNA. And then the type 3, you can just say with the type T, guess what that is? Type 3 will be tRNA. So we'll talk about rRNA and tRNA briefly in this video, and we'll talk more about it in the next video. All right, so, and also the rRNA, ribosomal RNA, is made in the nucleolus. So let me write that down. It's made in the nucleolus. Get our pen going there. The nucleolus, which is inside the nucleus. But mRNA and tRNA are made in the nucleus, right? So that's the difference is one is made in the nucleolus and the other 2 are made in the nucleus. And again, we'll talk about rRNA and tRNA briefly in this video and then more in the next video. So let's look again at how this is happening. So if we create this DNA, and we talked about this again, there is a promoter, and this is oversimplified, right? It's promoter, and that's where the ribosomal, the RNA polymerase, will begin. It'll start there. It's going to break apart the DNA, like I said before. And, I'm just oversimplifying it here with this. So this, if we're trying to create mRNA, this will be a type what? What do you think? It'll be a type 2, right? Because we're trying to create mRNA. So it'll be a type 2 RNA polymerase. There we go. And it's going to read it from the 3' to the 5' end. This is transcription, remember. This is transcription. Because it's reading the script. It's reading the code. And we did this before. So I'll just do it again. Why not? So again, let's just say, and this is the template strand. We talked about that in the last video. This is the coding strand. This is the template strand. This is the sense strand. It's also they call it the sense strand. And of course, this is the antisense strand. There we go. Oops. I made a fancy E there. So if we change the color so we don't blur ourselves, if we had let's say we had this as a T, another T, a C, it looks like a G, make it a G and an A, and it's going to read this. The T is going to become an A, C, C, and then U, remember, because of uracil. So this will be the mRNA. It's going to be generated, then it will leave the nucleus. Yeah, we showed this in the last video too. So here's the mRNA. It's now been constructed. It's going to leave the nucleus. It's going to end up in this cytoplasm over here. Here's the cytoplasm. It's the gel around the nucleus, and other organelles. And it's going to look for the ribosome. So we'll make this little weird kind of marshmallow cookie, you might say. That's the ribosome. And the mRNA is going to enter. It's going to enter the ribosome. So now we have mRNA entering the ribosome. We also have to talk about tRNA. So then the tRNA is going to be involved in this also. So as it enters, there is a tRNA has an anticode, codon that is, that's going to attach itself to the codon in the mRNA. So what am I talking about by codons? Let's look at that. All right, so here's a better picture of it. So here is the mRNA, as you can see. Right here, it says mRNA. Get our color here. And every three, and I'll draw them like this, every three nucleotides is a codon. Every three is a codon. And the tRNA has three, right? And it's the anticodon. And it's going to match up to the codon in the mRNA. On top here, this is an amino acid. And in the next video, we'll talk about how tRNA gets this amino acid. Basically, right now, with tRNA, is it's also made in the nucleus, but it's uncharged. It needs to be charged. And the only what that means basically is that the tRNA is going to gather an amino acid, and we'll talk about that in the next video. So as it enters the site, you can see that there's a top unit of it. It's called the large subunit. And then over here is a bottom one. It's the small subunit. So as it enters, there's three stages it'll enter. You don't see it here. There's an A stage as it enters. There's a P stage where it's going to produce these amino acids, this peptide chain. And then there's an E stage, means it exits. It exits without an amino acid. So that's basically what it's doing. Let's look at a better picture. So here you see the codon, right? So let's just say right here, there was, this codon right here as it enters the A site. Let's say that this was, let's just say anything, CCG. Well, the anticodon will have to have the corresponding nucleotide base. Remember, this is the anticodon on top of the tRNA, anticodon. So the C would be a G, C would be a G, and the G will be a C. And these two will match. As it comes in, it will match. As you see, it's coming in here. It'll match, and the amino acid will be released, and it'll create this chain. And again, we talked about peptides, and we talked about proteins. So all amino acids are proteins. They're just these little balls. They're not really those balls, but this is oversimplified. And so if you have a small chain of amino acids, a small chain, it'll be, put an arrow, a peptide, right? But if you have a larger chain, the larger one will be a protein, right? And there's more to this after the peptides are made and the proteins are made. There's further synthesis involved, but we'll talk about those in another video. So again, we're going to be talking about the tRNA in the next video, but again, it comes into the A site. It matches with its anticodon. It matches the codon of the mRNA. It creates these amino acids like popping out like bubbles, right? Or they're popping out like gumballs. And then it will exit. So we have the A site, the P site, just think about it as the production site, and then the E site as it leaves, it exits, right? So EPA, think of that. All right, and with anything in a factory, and you can think of this as a protein factory, the tRNA needs to know when to start. Just right, remember when we talked about the RNA polymerase, they need to know when to start, and a promoter started them. But in this case, they need a specific protein to help them start, and there is a start protein, which we'll talk about. It is called AUG, and I'll bring up a codon chart. And AUG is methionine or methionine. And here's our chart. So there is a start codon, AUG. Remember that. Think of August. Some people start school in August in some parts of the country, right? Get our color here. We'll use red. So if we want to know what AUG is, we start in the center. Here's A, and we go down from A, find U. Here's U, and then find the G. There it is, and you can see it's MET. MET is the code for an amino acid called methionine or methionine. I'll just spell it for you. I've heard people say methionine, or I've heard people say methionine. Whatever you like. Your teacher will correct you. I've had teachers in the past say methionine. I've had professors say methionine. So which one is correct. Great debate. So that's the start codon. So if we go back, let's go back. So if we here, let's say that this was the start codon, right? This would be methionine. And let's go back there. Right, so let's just say right here, we put AUG, methionine, August. And that is where the tRNA knows where to start. So let's say then it created over here, like we said before. Let's look at the chart and let's just say it created something. All right, so here's this tRNA, and let's say over here the tRNA had, well, let's say over here, it's reading this codon over here. Let's say, again, we had the start codon, which is AUG, so it started at AUG, A, U, and G, you get MET, thionine. Let's go back. All right, so let's say this was the start protein. And let's say over here that the tRNA now is going to, let's say over here we had, let's say it was C, cytosine, guanine, and uracil. And so the anticodon over here would have a GCA, right? That's the anticodon. And then out will pop, this is alanine, right? ALA is alanine. So that is the code for alanine. Let's look at it. Let's go back. So we first look for G. Here's our G in blue. Then we look for C. There's C. And then we look for A. There's A. And you see we have alanine. Alanine. Now let's look for another one. Let's say you have lysine, right? So for lysine is AAA. So we have A, A, A. And there's other codons to make lysine also. You can have AAG, AA, well, just AAG or AAA actually, right there. And the thing about lysine is it's an essential amino acid. We talked about that in previous videos. In other words, with an essential amino acid, the body doesn't make it. You need to get it from food. So again, just reading the chart, where would you find, let's say, I'll just write any one thing here. What about CAC? Where would you find CAC? All right, so let's say the anticodon was CAC, CAC. So let's get the pen going here. Here's C, then we go to A, then we go to C, and we have this, histidine. So histidine is the amino acid. So let's go back. So if the anticodon is CAC, right? What about CAC, histidine? What would be on the mRNA? So let's just say these are the three. This is a codon here. So this would have been G, U, G. GUG would have made CAC, and then you would have had histidine pop out. So you can see again, just a brief review here. We have the A site, the P site, the E site. tRNA is going to come in. Its anticodon is going to match the codon from the mRNA, and these peptide amino acid chains are being created. So you have, and peptides will be created, polypeptides. So now we're going to talk more about tRNA in the next video. tRNA gets its shape because it kind of looks like a T. We'll draw it somewhat over here. There's this kind of a weird shape to it. And I'm not the greatest artist, but we'll just, basically it has its shape like this, like a T. And we'll talk about it. That's why it's called tRNA. Well, not just that, it's also transfer RNA. It's transferring the information. We'll talk about that in the next video. And don't forget the start codon is AUG. But by the way, there also has to be a stop codon. Almost forgot about that, right? Just like we need to stop the video because it's getting a little too long. Last thing is there is a start codon, but there's also get our pen right here, a stop codon. And there's many stop codons, but there's also UAA is a stop codon. Where can you find it? Well, here's U. Then you go to A, and you go to A, and you see here it's a stop codon. In other words, it'll stop. So it's great. This factory inside of us is amazing. All right, next video, we'll talk about tRNA.